This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 902, recorded on May 20th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today, right here at the incubator, Rich Condit.
Hello there, Vincent.
Hello,
here we are
Rich.
together again. This is great.
Now, you may be wondering why Rich is here. We had an opening of the incubator yesterday, and Rich came, as did our second host today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. Let's see. Looking out the window, it's a little grayish out, but the weather is bordering on summertime, and I can tell you that the weekend is going to be a scorcher. And we're going from 2 feet of snow on the ground to here come the daffodils. Forget that. There comes the crocuses. They've already croaked. And then next thing you know, we've got summertime plants coming and going. The birds are terribly confused. They are confused, by the way. And then there was a wonderful report just recently on NPR saying the best place to go to watch birds migrate is Central Park in New York City. So, Rich, if you get a moment, get out there and look for the vermilion flycatchers. Come all the way
Well,
from Mexico.
I may have a moment tomorrow. I'm going to be around tomorrow for a little while. Today I went down to. I've been outside. It's by the way, it's can't be much different than where it is where you are, Dixon. But the Weather Channel says it's 66 in New York, and that's about right. And it's been sort of partly cloudy, so sometimes it's sunny and warm, and other times it's a little chilly and cool. But I've been out in it. I went to Ellis Island and Liberty Island today.
Great.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see everyone. It's been such a long time since seeing you all last night, or seeing some of you last night.
Brianne was also here yesterday at the incubator opening.
Yes, and the Weather Channel says it's 71 in Madison, and also tells me that we are under a tornado watch, which I was not aware of till this moment.
Is that interesting? Isn't that interesting?
Wow. And from Ann Arbor, Michigan, Kathy Spindler.
Yay.
Hi, everybody. Here it's 84 Fahrenheit, 29 Celsius, partly cloudy.
So, Rich, you see I have no windows and can't see, but It's 19C, and Rich's weather is the same as mine today. How about that?
How about that?
Yeah, it's really something. So, as everyone knows, I talked about it last time. I went to Zurich to get the Richard Ernst medal, and I wanted to tell you two things. First of all, here's the medal. It's a solid. Let me see if I can see what I'm showing you. Yeah, it's a solid gold medal. So Richard Ernst did the first MRI on his own head, so that's what that is. And then on the other side, it has my name. So that's really cool. You want to see that? Yeah. And this is for Kathy. I'm going to go back to my camera for a moment. So there's a French horn quartet in between different parts of the ceremony, right? And one of the they're all chemists from the department. And one of the players wrote a piece for this occasion, right? The Richard Ernst lecture. And look at what he called it. Can you see that?
I'm not quite in focus. Ernst?
Yeah. So it's going to be, I'll have to read it to you, but it's called Ernst Fanfare 2022 CD 155.
Oh, perfect.
Isn't that great?
That's really good.
And he said it also starts with C and D. Those are the first two notes.
Oh, very good. So now you have to explain to some of the listeners what the CD155 significance is.
I will. So CD155 is the name for the poliovirus receptor, which was identified in our lab many years ago. I just thought that was lovely. S.D. Hogan is the author. So that's the whole thing. And I guess each line of music is for each horn, right?
Yes.
That would be how it works.
Mm-hmm.
Isn't that great? And he gave it to me. I have a picture with him as well.
That's great.
Did you see this yesterday?
Yeah, I did.
This is very cool.
Very cool.
It was such a nice honor. I had a great time.
So this is like solid gold?
They just said it was gold, and it's really heavy. I don't know.
It's really heavy.
Well, I just read, why is it that the Olympians bite their medals, and it's to prove that they're gold, because if you bite it, your teeth marks will show if it's true gold. But that's not the case anymore for the Olympic medals.
I was going to bite it. So it shows teeth marks if it is gold. I'm not going to.
Supposedly, yeah.
I don't want to put teeth marks in my medal. No.
Yeah.
All right. So
there was one other thing. We did have a little bit of follow-up that indeed the Ernst L. Winokur that I thought of as the adenovirologist was the one who also won that prize back in the day.
Okay. Wonderful. Anyway, I wanted to thank Serena Riniker, my host, for taking care of me. She met me Sunday and walked me around Zurich, and we went on a boat ride. She picked me up on Monday, got me a rapid antigen test, so in the square in Zurich, they set up this structure, right? And you can go on two lines, PCR or rapid antigen. And they took my email and my license, and I took an antigen test, and it was free. You know, you can't beat the Swiss. And when we got to her university, ETH Zurich, I checked my email and I had the result. And I had to upload that to my boarding pass to get home. So that was quite a relief. So thank you, Serena, for doing all that. I really appreciate it. All right. So before we get to our snippet in paper, I wanted to hear from Rich a little thought about these cases of monkeypox that the news is going crazy over. And in fact, there's even one suspected one here in New York City. There was one in Massachusetts yesterday. What's up with that, Rich?
Hey, I don't know. Because it's all the two things that I've read that are of most interest to me. One is the ProMed Mail link that you have in here, because I trust that as a reliable source. And the other was Ed Young had a piece in the Atlantic. Mm-hmm. The Atlantic, in addition to doing its magazine, has an online presence, and they publish short pieces. And one of our, who is that? Justin Moore, Kathy, who sends us stuff now and then, sent us a link to an Ed Young piece that's actually quite good. But what I'm missing is some sort of summary of the distribution of these cases. There's some in the UK, right? Mm-hmm. Mm-hmm. There's at least one in Canada.
Yeah, there's a cluster.
Is that on the ProMed?
I think there's some maybe in like Spain or Portugal or somewhere else in Europe.
Right.
There's a cluster around Montreal. Yeah.
And so,
where do these originate, Rich?
I don't know. I mean, the monkeypox is most common in Africa, and certain regions of Africa, and I should be able to be more precise on that, but I'm not. Zaire, one of the strains is named Zaire strain, I believe.
I'm pretty sure it's Central Africa.
Right. And typically, it's been around forever. Monkeypox, as with many of these pox viruses, is a misnomer. Its native host is thought to be rodents, and they pass it around in fashions unknown, or at least unknown to me. I think not clearly defined. And occasionally humans come into contact with these animals and can pick it up. It requires the transmission to humans and among humans requires quite close contact. So either direct contact, and it can infect you through mucous membranes or fresh wounds in the skin, or if some other animal is actually has an infection that has respiratory activity, it can be shed in respiratory droplets. But you got to be really close to pick up enough of a dose. So there are outbreaks going on periodically, reasonably frequently in Africa, and they are by and large controlled. Most of the time, they burn themselves out because human-to-human transmission is not very efficient at all. The R0 is not well defined, but it's thought to be less than 1. So that's insufficient to efficiently sustain human-to-human transmission. And I looked it up last night, secondary attack rate of 10%. Okay, so you got a 10% chance of catching it if you're in like a family situation, fairly close quarters with somebody who's infected. So both of those combine to not very transmissible.
I'm going to take your coffee cup away because you're tapping it on the table, dude. Okay,
don't
sorry.
take it away. Whatever you do, don't take it away. I want that.
So the clusters are in Britain and Portugal, Spain, Sweden, and Italy. And the ones in Britain and Canada are mostly men who have sex with men.
So that's the weird thing. The unusual thing about this is that there seem to be a lot of these popping up in the same period of time in multiple different places. And it's hard for me to believe that there isn't some common source at the beginning of these, but it's not been identified. And nobody has a clue. I don't have a clue.
So, Rich, what about the long-term consequences? Scarring?
So this is a close relative of smallpox. And so the pathogenicity is close enough so that the smallpox vaccination will protect you against this.
Really?
Distant enough so that the host range and pathogenicity are different than smallpox. So smallpox infected only humans, as far as we know. There was no animal reservoir, whereas this has its natural reservoir is probably rodents, and humans are an incidental host. It doesn't transmit as well as humans. The disease has a milder pathogenesis, but I think the pathogenesis is similar, where you can catch it as a respiratory infection, and it will replicate locally and then establish a viremia. I'm talking now about mousepox and smallpox, but I think the same goes for monkeypox. Infect internal organs, establish a secondary viremia, and ultimately erupt on the skin. And so what you experience is a period with headache, nausea, malaise. You feel just really crappy. And then ultimately, there can be lesions that erupt on your skin, a rash of one sort or another, pustular, quite characteristically. And it clears up within a couple of weeks. Case fatality rate is between 1 and 10%. Okay. And that's actually pretty high. If you consider that, what's the case fatality rate for SARS-CoV-2? I even forget, like 0.2%, 0.3, between 0.2 and 0.4%, something like that. So 1% is pretty high, but typically not a lot of people get infected. It's not clear. There are a couple of different strains of monkeypox identified, some of which have a higher case fatality rate than others. And I have not seen any genetic information on this stuff yet. So I don't know what strain is going around or whether everybody's got the same strain. Okay, so a lot of unknowns at this point.
CFR of COVID can vary depending on how old and so forth, but it can go up to 6% or more. Yeah.
For this virologist, the monkeypox outbreaks kind of pop up every now and then. So I just, I think it was in PubMed or Google, I just put monkeypox Wisconsin and got that there was the outbreak in 2003 associated with prairie dogs. But I remember one from the late 1990s, also somewhere in the US, because I remember we talked about it in my undergraduate class. So they just come up every now and then.
Yeah, the thing that I also thought about that prairie dog associated outbreak that's related to exotic pet importation. I always talk about that one in my emerging infectious disease class. And the thing that I sort of wondered was exactly how long it took us to figure out that there were these sporadic monkeypox cases, and to tie it back to these prairie dogs, and kind of to think about how that might link up to the timing of what's going on right now.
Yeah, my guess is they'll have this figured out within a week or two. There'll be a whole bunch of genetics. There'll be some some history on the individuals that are infected, and it should be interesting. It's not something, of course, we get it wrong, not infrequently, but it's not something that worries me. Okay, and I think it's in particular with respect to the U.S., but I think a lot of this applies to other countries as well. In the U.S., in response to the 9/11 attacks, that's what precipitated the whole biodefense program in the U.S. And as part of that program, they identified pathogens that were they call it Class A, or high-risk pathogens, that were sort of the several most wanted of pathogens that might be good bioterror weapons. And the top of that list is always smallpox. And then the mandate became to create a vaccine and identify antiviral drugs. A vaccine and 2 antiviral drugs for each of these. And that's been done for smallpox. So we already had a smallpox vaccine, but there was more put into that. We're now on the 3rd generation. And so there are a variety of different smallpox vaccines that have been stockpiled, millions of doses in the U.S. And there have been developed 2 drugs. One, Tecomat or something like that. I forget what it is. I keep calling it ST-246 because that was its original name. But that's actually been approved by the FDA, and stockpiled in millions of doses, and is extraordinarily effective. And the vaccine for smallpox, the drug and the vaccine, although they were developed for smallpox, work with monkeypox. So, and there's another drug as well, cidofovir, that works. So, and we have a lot of experience with how to deal with smallpox infections or poxvirus infections in general. So even if this were a somewhat unusual monkeypox infection, I'm totally confident that we could deal with it in short order. But I don't think it'll ever amount to much.
Now, I remember you saying that this became more common after we stopped vaccinating against smallpox, right? Because you can get protection.
Correct. With
the smallpox vaccine. Yep. And also, you said that monkeypox is a misnomer because that's not the reservoir, right?
Correct.
Okay. It's rodents of some kind.
Yep.
Okay. Well, probably next time we have a TWiV, we'll know more. There you go. Dixon, you good with that?
Sure. I was also going to ask something about the rodent reservoir, if you wanted to call it that. Are these asymptomatic and you can't tell by looking, obviously, whether they've got it or not?
Actually, Dixon, I don't know. I don't know.
All right. And also a long-lived virus on fomites, which are transmitted because smallpox can do that, right? The blankets that they gave the,
right.
as I recall, the Huron Indians actually extincted them.
I presume that the same goes for monkeypox. Can be transmitted on fomites.
So maybe that's how it gets around rather than for the actual host itself. Who knows?
Who knows?
Who knows?
We'll find out.
Great.
Okay. Our snippet today is a scientific report that's one of Nature's journals. Excretion and viability of SARS-CoV-2 in feces and its association with the clinical outcome of COVID-19. It's from a number of groups in Spain. The first author is Cristina Serrada Romero, and the 2 corresponding authors are Francisco Pozo and Javier Sanchez Cespedes. Anyway, so we have mentioned multiple times this idea that, you can find SARS-CoV-2 nucleic acid by PCR in feces because we swallow mucus and it passes through. But the question is whether the virus can replicate in the intestine and therefore might be transmitted fecally orally. And other coronas are excreted. Other coronaviruses are found in feces of infected patients, and they're infectious, actually, which is amazing because the stomach is a harsh environment and the small intestine. But for SARS-CoV-2, we're not sure. And they say 6 studies, which include 371 adult patients, COVID-19 patients, reported detection of the virus in stool and rectal swab samples with this is by PCR, with frequencies ranging from 15% to 81%. And some studies have reported the presence of viable viruses in stool samples. And a patient recently with rectal surgery that was performed 3 days before COVID onset, they claimed direct evidence of replication in intestinal tissue. And that paper, they said, here's a quote from it: coincident with rectal tissues of surgical specimens testing nucleic acid positive for SARS-CoV-2, typical coronavirus virions in rectal tissues were observed under electron microscopy. Moreover, abundant lymphocytes and macrophages, some SARS-CoV-2 positive, infiltrating the lamina propria, were found with no significant mucosal damage. So they say, all right, what's going on? So they have a cohort of 62 patients, median age 59 years of age, 60% of them male. 19 had some chronic underlying condition. And they collected stool samples from these patients at admission and during follow-up up to 4 months after the onset of symptoms. And so they looked first by PCR, and 43%, that's 27 out of 62, had viral RNA in feces by PCR. The CT values, the cycle threshold values, in the first 2 weeks were ranged from 24.5 to 39.6, in the first week, and from 27 to 39 in the second week. And they also have CT values for nasopharyngeal swabs, which range from 17 to 30 and 11 to 37 in these 2 time periods. Okay.
Yeah, and we should say something about the numbers here, because the numbers start to tell you a little bit of the story if you're used to thinking about RT-PCR. So CT values are the cycle at which the sample becomes positive after amplification. A larger number means it took more cycles to amplify, which means that there was a lower amount of starting material. So a low CT actually means a lot of virus. And if you look at these numbers, many of these numbers that they're listing for the stool samples are on the high side.
Yeah.
And when they give, which means there's very little virus. And the comparison is to those nasopharyngeal swabs, which are very much lower. So that starts to say, okay, yes, they are getting positive stool samples, but those stool samples do not have the same amount of nucleic acid. They have less nucleic acid.
They say in the discussion that if the CT is greater than 24, you have significantly reduced infectivity.
Right.
And so then they tested these for infectivity. So they take these stool samples and they inoculate cell cultures in the laboratories, and they found cytopathic effect in 24 of the samples at 77%, 3 to 7 days later. And then they took the supernanes of those and passed them again, I think, subculture, and then they did an RT-PCR for SARS-CoV-2. None of them were positive. So despite getting CPE, cytopathic effect, must be some other virus in there, right?
Exactly.
And none of these are PCR positive for SARS-CoV-2.
Oh, could be CPE from virus, or could be just CPE from nasty stuff in the stool sample.
Yep. Nasty's in the stool.
Haven't earlier studies looked for the virus in sewage, and then didn't they take that to the next step and try to see whether the virus particles were infectious or not? Isn't that an older set of
Well, they've certainly looked by PCR in the sewage. Yeah, I'm not sure they look for infectivity.
You could look at sewage disposal as a way of initiating new infections downstream from wherever they're being shed, and so it's important to know whether or not these CPE or effects are actually due to the virus that they were looking for to begin with, right?
Well, the PCR says it's not SARS-CoV-2, right?
That's what it says.
Yeah. So, I mean, our friends here in New York who do the sewage PCR who we've had on, they said they haven't been able to look for infectivity because they need containment that they don't have. And I don't know whether others have looked or not.
That's right. They pasteurize it.
Oh, that's right. They pasteurize
when they get it immediately.
That'll do it.
Pasteurization works.
It does work. It does work.
So that's an excellent experiment that you suggest, though, Vincent, because if they don't do that, then they're obviously not thinking of the more obvious experiments to do. It seems that would have been one of the first things they would have tried.
The problem with this experiment, all right, so it's negative. They say, no, we don't find any evidence for replication. And the people where we saw PCR positivity, their clinical picture was no different from anyone else. And so they conclude that, it's probably not transmitted this way, but they do admit they have a small sample size. But I would say maybe you couldn't detect infectivity because, as Kathy says, they're nasties and feces. It might inactivate any virus that's present. So it could, in principle, replicate, but you could never tell because, and so I think the experiment might be to take feces from an uninfected person, mix it with different amounts of SARS-CoV-2,
absolutely.
and then measure infectivity and see
That's right.
if you get good correspondence with untreated or not. So that would be fun to look at, I think.
The classic spiking experiment,
Spiking.
as Vincent has put it in here.
How about just in physiological saline or water, just pure distilled water? Those experiments must have been tried some time ago, no?
What do you want to do?
Just take the purified virus particles and put them into a pristine environment first to see whether or not how long
that would be your control.
in nature.
Sure, that would be your control. You do a titration of your virus stock, and then you mix it with feces, and you say, is it exactly the same titer? Does it go down tenfold, or do you never see infectivity? Right. I mean, the paper I mentioned that they cite where they see particles by electron microscopy, I can believe that now and then maybe a particle is made, but if it's only a few, it's not going to contribute to transmission. So you need an infectivity experiment to figure that out.
How many times has TWiV asked for the spiking experiment?
Quite a few.
A lot.
One of these days we're going to get one. I'm looking at this. I'm looking at this paper that they referred to that is referenced by saying that there is evidence for virus replication in the intestine. And this is some of the best evidence I've seen for this. They got electron micrographs that are reasonably convincing.
But we don't know if it's SARS-CoV-2, right?
Well, they've got immunostaining with SARS-CoV-2 antibody in intestinal tissues. And, there's little red dots all over the place. Looks pretty good to me.
So is it multiple cells or one cell?
Must be multiple cells as I'm looking at this image. Here, check it out.
Yeah, it's a single patient, though, too.
True.
Yeah.
The other thing, back when I was mentioning the nasties in the feces, not just the nasties doing something to any other viruses or virus infectivity, but just nasties being nasty to your mammalian cells in culture.
Yeah.
Right.
We've done experiments where you put mouse urine onto cells, and that can be pretty nasty too, unless you dilute it out a lot.
This is true.
I'm sorry, Kathy, we got a title now. Nasties in the feces. It's great. It's just great. But yes, you're right. It's not just for the cells that show CPE, but it's to inactivate the SARS-CoV-2. So you could have production of virus in these cells, and you would never detect it. And if you don't ever detect infectivity in feces or very infrequently, then it's not a transmission risk, right? If you don't have any infectivity. Because the last time I heard, SARS-CoV PCR positivity does not transmit on its own. You need infectivity. What did you say, Dixon? I'm sorry, interrupted you.
No, I was just agreeing with you. That's all.
So anyway, this was a very quick study that I wanted to tell you about because I don't think we quite have an answer yet. I mean, this is a small study, but I wish they would spike it.
Right.
Spike it. Okay.
Dear people with BSL-3s, please do the spiking experiment.
So this experiment to take fecal samples and infect cells, that must be a BSL-3, right? If there's potentially SARS-CoV-2.
I think so.
Yeah.
Yeah, I would think so. So they could do the experiment, yeah. All right. Any other comments before we move forward? All right. Now, the paper I thought was really amazing. It's a PNAS paper. And let's see, when was it published?
It looks like last week, May 16th.
Yeah, okay. So the risk of COVID-19 death is much greater and age-dependent with type 1 interferon autoantibodies. And I'm looking for the authors. And I say, how come there are no authors on this paper? They're all stuck at the end because there are billions of them, right? Holy cow.
Yeah, so I'm looking at a version that is not the PDF. I'm just looking at an HTML version, and it has 3 authors listed. It actually has 4 authors listed, but there's 3. And then it says plus 194, and then it lists the senior author.
Because in my version, the PDF, there's no authors up front. They're all down below. And then the affiliations go on for another 2 pages. It's amazing. And I'm not even going to try and...
But I do like the plus 194.
I mean, is there a first author here? Let's see. The first author is Jeremy Monry, right?
Right.
Mm-hmm. And the second author is Paul Bastard. And he published a paper earlier in the pandemic about type 1 interferon autoantibodies and SARS-CoV-2 that I found really fascinating. So I think this is sort of a follow-up of that.
I'm looking through the authors here, and on this paper is Jean-Laurent Casanova, his work is all about finding genetic correlates with severe disease. And
where's he?
He's Rockefeller. We also have Charlie Rice on here. Okay. Anyway, I find actually the introduction of this paper to be almost as important as the data themselves, because they summarize a lot of good stuff. So let's talk about it. So the course of infection with SARS-CoV-2 ranges from silent in about 40% of the cases, which means, asymptomatic, to acute respiratory distress in 3% and death in about 1% of cases. All right. And they say age is the strongest epidemiological predictor of COVID death. It doubles with every 5 years of age from childhood onward. Wow. Dixon, don't get COVID.
Well, that's too late. I've got it in December, but I didn't have type 1 interferon autoantibodies, so I'm okay.
Well, sounds like it, or other things, because as we'll learn, as you age, there are other things that also.
I don't want to
know. But Dixon, you don't have any of them. So did you survive? That's what I would like to know.
I presume so. You be the judge. My persona showed up, but I'm not sure it's the same one that I had before the infection.
I think you're pretty similar.
No. The only thing I find has altered after that is my perception of saltiness in food. And yesterday's wonderful party that you threw, I could not eat most of those prepared meats. I tried a variety of them, and
they're too salty.
Much too salty. I mean, overly. I think my ability to detect that has become exaggerated. How's that?
Interesting, because usually it's the other way, right? You go.
Well, you don't know, because
it could be.
A lot of this happens after the receptors have been stimulated. What happens between there and the place where it's translated in your brain somewhere? And that's tough to know about. And these viruses obviously affect those regions. So
okay,
I'll let you know if it returns to normal or not.
Interesting. All right. Anyway, so age is the biggest risk factor for death. and men are at greater risk.
The older you get, the more likely it is you're going to die. I don't think that's true. That doesn't relate to anything else but just getting old.
Okay, and then beyond that, inborn errors of type 1 interferon immunity are important determinants of death.
Sure.
So the COVID human genetic effort has shown that type 1 interferon is essential for protective immunity, and inborn errors so there are sensors of the interferon system like TLRs, toll-like receptors, and there's a study that shows that changes in TLR3 gene can contribute to life-threatening COVID in some patients. Mutations of 8 genes have been found in 23 patients with COVID, and 4 of these patients, unrelated, have autosomal recessive deficiencies of IFNAR1, the type 1 interferon receptor, or IRF7, an essential transcription factor for the production of interferon. There's also an X-linked TLR7 deficiency, which is seen in 1.8% of the cases of life-threatening COVID. So there are a number of previous studies that have pinpointed deficiencies in interferon and interferon sensing. Anything I miss, Brianne?
The one thing that I would point out, and then I want to see what Kathy wants to say, is that the difference in risk with men and women is at least partially tied to this type 1 interferon genetic aspect because of some genes, including TLR7, like they mentioned here, being X-linked.
Right.
So that's obviously not all of that difference in risk, but at least some of it has to do with a surprising number of immune genes, in fact, are X-linked.
So
I was just going to say
how many
that.
other viral infections?
Dixon, let Kathy go. Sorry.
I'm sorry.
That's all right. I was just going to say that, to me, this first paragraph of the introduction is talking about genetic reasons for why the interferon response is altered and has effects. And the next paragraph that we haven't yet gotten to is going to talk about sort of, in a way, biochemical or immunological reasons why the interferon response is playing a role. So there's sort of, to me, there's genetics and there's biochemistry. And I was just going to also say, when we talk about inborn errors, your brain should immediately just be triggered to think, oh, okay, genetics.
Dixon.
Just a note, could you think of any other viral infections which would be lethal if type 1 interferon were not effective?
Well, so that is part of the thing that gets so fascinating to me about this story, more so with the part with the second paragraph that Kathy mentioned that we haven't gotten to. Because what's really unique about the patients that they talked about, both actually in some of the papers that are described here in this genetics section, but also the patients that they talk about in the next section that have an issue with the interferon response, is that those patients actually were not known to have interferon deficiencies before they got severe COVID. So it seems like they were okay with many other viral infections. And the severe COVID was sort of the first sign that they had this interferon issue. And that's something that the Casanova lab has looked at with a number of different microbial infections and immune genes.
But there are some inborn errors of genes associated with TLR3 or 7, I don't remember, which predispose to severe herpes encephalitis, right?
Right, yes.
They're probably different from the changes that we're seeing here, which is really interesting, right? Which tells you that there's some virus-specific changes.
Yeah, I mean, it's so interesting to me to realize, to see how this shows that the immune system has a lot of places where it can be rather redundant in response to certain microbial infections, but very specific microbes can lead to problems with a very specific defect in some part of the immune response. And we don't really understand why that is.
Makes sense, right? Because if you had one defect that predisposed you to all sorts of viruses, you would never survive. species
exactly.
would be wiped out.
Yeah.
Okay.
I mean, if you just think about what we know about the genetics of susceptibility to viruses, that it oftentimes can be just to one virus and one particular gene with an immune function.
Which is something Kathy has worked on in her career.
Mm-hmm.
Okay, paragraph 2, which everyone's very excited to get to. So, and that's the topic of this paper. The authors have also found autoantibodies. That is an antibody against something in you that neutralizes high concentrations of interferon alpha-2 and or interferon omega in 10% of patients with critical COVID pneumonia, but not in individuals with asymptomatic or mild infection. And that finding has been replicated in 14 other cohorts. That's good. That's impressive.
Yeah, it really is. I remember when that paper that they cite here, reference 12, came out, and reading it and just having my mind absolutely blown by these ideas of these antibodies against interferon that were neutralizing interferon, and basically leading these patients to have no type 1 interferon response and thus be susceptible to severe COVID. I was just blown away. And the thing that becomes really fascinating, again, if you would ask me before I read that paper, were there such things as anti-interferon autoantibodies in patients, I would say, oh no, I've never heard of that. I don't think that exists. That would be a huge problem in patients. And these people were not sick beforehand. They seem to be okay. There has been a little bit of debate about when exactly the antibodies were present, because you don't have samples from these patients before they got COVID to show that they had the antibodies all along. So there's been a little bit of debate on that point. But just the idea that there can be these anti-interferon antibodies, and you could be walking around perfectly healthy with these antibodies, and then you could get infected with SARS-CoV-2 and have a severe outcome, was just fascinating.
One of the things they suggest is maybe we should screen certain people for these antibodies, right? Which just seems like a good idea.
How common is this thought to be?
I had not heard of it until their reference 12 paper.
And it also suggests a way of interfering with the interferon's interferon. And that is, if you gave these people steroids to lower their antibody responses and then gave some type 1 interferon in addition to that, you might be able to overcome the effect that autoantibodies were having on the immune response.
Yeah, the other part that was really interesting about it is that there are actually multiple subtypes of type 1 interferon. So there's interferon alpha, and there are different numbers, alpha 1, alpha 2, all sorts of different letters up to interferon omega. And it was a specific type 1 interferon alpha 2, and interferon omega, that seemed to be the ones that were important here.
It's amazing.
So, Rich, to answer your question, in the discussion, they talk about the increasing prevalence of autoantibodies in the general population with age, around 1% under 70 years of age, and over 4% over 70 years of age. So 1% to 4% by the time you turn 70.
There's this age thing popping up against.
But isn't that's broad? That's not specifically the interferon ones. That's any autoantibody.
Autoantibodies, okay, but still.
But then they go on to say the proportion of deceased patients with these autoantibodies is stable across age categories, around 15 to 20%. So that's not so much age-related when you look at people that die.
Okay, moving on. So they also found autoantibodies that neutralize lower concentrations of interferon alpha-2 or omega in 13% of patients with life-threatening COVID, 18% of patients who died of COVID. And they also found in 1.3% of patients with critical COVID, they have autoantibodies against interferon beta without autoantibodies to alpha-2 or omega. It's just amazing, right? That you would be so specific. And then as I think we just said, the prevalence of autoantibodies against alpha-2 or omega in the general population increased with age from 0.18% and And for the low concentration and 1% for the higher in individuals 18 to 69 years of age to 3.4% and 6.3% for over 80. But the prevalence does not increase against beta. You just said that, I know. Yeah. And then they have a table here which has at least 12 lines of evidence suggest that autoantibodies are strong determinants of COVID-19 death. And so there's evidence and examples for each and references for all of them. And so what they do in this paper is to use previously reported data on this to estimate the relative risk of death, RRD. So what's RRD? Can someone, I think, Brianne, last time you explained it, right?
I think I might have, but I hadn't prepared to explain it right now.
I'm sure you can remember now.
Yes.
You don't have to tell us how we do it. Just tell us what it means if you get a number.
So my recollection is if you have a relative risk of death of 2, that means you are 2 times as likely. So twice as likely to die. And if you have a relative risk of 0.5, that means you are half as likely to die.
And you're comparing it to a population with different conditions, right?
Yes.
Like with or without autoantibodies.
Right, exactly.
Yeah. Okay. So that's what they did. They estimate this relative RRD for people carrying autoantibodies for these type 1 interferons. And I just should tell you that they use Firth's logistic regression. Okay. They have 1,261 patients who died of COVID and 34,000 individuals from the general population, and they have samples from them before the pandemic. So they go through some work to show that their numbers are probably okay, but let's just go to the numbers. For autoantibodies neutralizing low concentrations of alpha-2 or omega, they looked at the association of these antibodies with death. So let me just get to the numbers. Oh, where is it?
While he looks for the numbers, just one thing that I do want to point out, I really like that they looked at patient samples from before the pandemic where they had them and some individuals from the general population, because that does address the concern that came up with their first paper about whether the antibodies were before or after infection. And so they show some data here that these antibodies were pre-existing and the patients were perfectly healthy with them before COVID. And so I really like that they did that.
All right. So this paragraph is basically saying how many patients of each level of interferon they had and which type. So then the next is the RRD for individuals carrying autoantibodies neutralizing low concentrations of alpha-2 or interferon omega. Overall, for these individuals, the RRD goes from 17 before the age of 70 to 5.8 for individuals over 70. All right. And so Kathy wrote in the show notes, we should probably explain this now rather than wait for the discussion, because otherwise people are going to be confused, as I was. I'm like, wait a minute. Brianne said it's the risk over. How can it go down? I would assume it's going to go up. But I guess, Kathy, the idea is that there are other factors that, so the contribution of autoantibodies to interferon becomes swamped by other things that happen as you get older, right?
Right. Most easily understood by just saying comorbidities. But yeah, basically it goes from 17 to 6 by the time you get to 70. So it's the relative risk goes down from 17 to 6. Why does it go down? Well, because compared to other things, when you're over 70, the risk from autoantibodies has gone down.
Right. So in the younger individuals, if you have antibodies, that's going to give you a lot of risk. That's going to be a real problem. If you're older, there were other things that are also giving you risk, so the antibodies don't contribute as much.
So the 17 means you're 17 times more likely to have severe COVID if you have these autoantibodies neutralizing low levels of alpha-2 or omega.
Right. Compared to someone else who does not.
Yeah.
That's a big number.
It is.
Yeah.
Yeah, I would like to know if I have autoantibodies. That could, dictate treatment. The presence of these autoantibodies neutralizing both alpha-2 and omega was associated with the highest RRD, 188. That's even bigger number.
Yeah, that's huge.
188 for people under 70 and 7.2 over 70. That's a huge decrease. So these comorbidities really make a difference as you over 70. That's amazing. Okay, so the conclusion is that having autoantibodies is a good predictor of severe COVID. Then they looked at the RRD for autoantibodies that can neutralize high concentrations of type 1 interferon. So, Brian, what is this low and high concentrations? What does that mean?
So I think that it has to do with kind of weird pop-up. So it's sort of how effective the antibodies are at neutralizing, and whether the antibodies you have would partially reduce your interferon levels or fully reduce your interferon levels. If you have antibodies that are, acting against only the low concentration, then perhaps you you might have some interferon still left. You might not remove all of your interferon, where with these antibodies, those that neutralize high concentrations, they're very effective. And so they are probably getting rid of all of your soluble interferon.
Okay. All right. So high levels, the RBD for people with autoantibodies alpha-2 or omega went from 64 before the age of 70. And by the way, there's a big range. The range is 38 to 101. So this is a bit noisy, I guess, before the age of 70 to 6.8 after the age of 70. So another big decrease. Whereas then people who have antibodies to both interferons have the highest 156 and 12.9 for those two age groups, less than 70 and greater than 80. So I guess that's higher than before, right? So for both, it's 156 and 12, and before it was 188 and 7.2. Not that different, right?
They're not that different. And I think that the numbers of people that they're looking at in some of these cases are not huge.
Not really big, yeah.
Okay. In particular, when they get to the part where they talk about people who have autoantibodies against both alpha-2 and omega, the numbers are maybe, the difference isn't as dramatic as I would have thought. And then I started to think to myself, well, how many people under the age of 70 who have autoantibodies against both of these interferons are they actually looking at here to get these numbers?
They do conclude that if you have higher proportion of autoantibodies neutralizing high concentrations is more deleterious with respect to severe COVID. They do make that conclusion, right?
Yes, exactly.
And then they also say high doses of autoantibodies against high doses of interferon beta had the lowest RRD before 70, which was 7, and no significant age-dependent association. So autoantibodies to beta it doesn't seem to be much of a problem, right?
Mm-hmm.
All right. Now they estimate the infection fatality ratio. And I remember, Rich, many years ago, you wrote
many years ago, like 2.
You wrote
that was many years ago.
It seems like forever. You wrote a blog post about IFR. Remember?
Yeah, because I was all
worked up,
excited. I was worked up. That's good term.
So, what is the infection fatality ratio?
That would be as opposed to the case fatality ratio, which everybody was talking about. Okay.
Rate or ratio?
Excuse me?
Rate or ratio? I think they're rates.
Okay, rate.
Yeah.
The case fatality rate would be the number of fatalities divided by the number of cases.
All right.
But I was worked up because, especially at the time, we had trouble counting the number of cases. And I thought, in particular, since there's a bunch of asymptomatic infections, a better number to have would be some sort of score of infections. And to me, that the best proxy for that is serology. You have antibodies that are a record of infection. So the infection fatality rate would be the number of fatalities divided by some measure of infections. Now, what do they use here? I don't know.
Yeah, I don't remember. So just for, since I'm starting to turn on my statistics brain again a little bit, it's been a long week. The relative risks were basically something like an IFR, or a fatality rate, of those with the antibodies divided by those without. And here we're just doing fatality divided by all those infected.
Yeah, I don't remember how they calculate.
I'm going to look it up.
You can look it up. Okay. So you look it up. We'll start talking about the data. So for all combinations of autoantibodies, the IFR in those with autoantibodies was much higher than the overall IFR. And they show that for some specific examples. Low concentrations of interferon alpha-2 or omega, it exceeded 1% and 10% for people over the ages of 40 and 60. The highest IFR observed for carriers was for neutralizing both interferon alpha-2 and omega, reaching 40.5% in individuals over 80. Infection fatality ratio, 40% in individuals over 80. That's really remarkable. And they say the values were similar for all other combinations of autoantibodies. The infection fatality ratio with autoantibodies was generally higher in male subjects than in female subjects, particularly in those with antibodies against both alpha-2 and omega.
So that also tells us perhaps there's something going on in males outside of the interferon predisposition.
Yeah. So that was low concentrations. Then they do the infection fatality rate with autoantibodies that neutralize high concentrations, and they get similar numbers. They range from 3% before 40 years of age to 68% in those over 80s. For those who have autoantibodies that neutralize both alpha-2 and omega. All right, so that is it. And they basically say that this is probably second to age in risk factor for severe COVID, having autoantibodies to these interferons, alpha-2 and omega.
All they say about the infection fatality ratio is, or infection fatality rate, is that they used the fatality rate upon infection with SARS-CoV-2 in the general unvaccinated population provided by, and give authors and a reference. Okay, so I would have to look up that reference. But it's an estimate, put it that way. Okay, and my guess is there's math involved.
Yes, indeed.
I'll even look up that reference. Sorry, go ahead, Kat.
I like to do an exercise sometimes where I say, if you could only show one figure out of a paper, which one would you choose to, give the most important or the take-home message or something? And I really like figure 3 in this one, where they plot the infection fatality rate on the y-axis versus age in years for different clusters of age groups. And the take-home message is that there's a higher infection fatality rate for carriers of autoantibodies to interferon alpha or omega. And then they show that there's a slight difference between males and the total males plus females, I guess, is what that bottommost curve is. But it's quite striking.
Very striking.
And I think this is open access, but I don't know that.
It is.
Yes.
Okay. Yeah, I got it on the train this morning on just the Wi-Fi. Yep. So they say, Brianne, that patients with autoantibodies against alpha-2 have been shown to neutralize all 13 interferon alpha subtypes, rendering any potential redundancy inoperative.
Sure, but we do of course have things like beta.
Yeah, right. But among the alphas anyway, that's
right.
Maybe because we had kind of inferred earlier that alpha 2 is critical for this, but maybe not so much alpha, right?
Right.
It's redundant anyway. Yeah.
Which is just really interesting because interferon alpha, all of those many types, and interferon beta use the same receptor.
Yeah, they do.
IFNAR, the interferon alpha beta receptor.
Mm-hmm. And
so it becomes really interesting to think about the fact that this may be, particularly specific to interferon alpha and maybe less so for beta, and think about how that signaling could happen.
Yeah. The other thing they note in the discussion, so autoantibodies against interferon beta were less common and associated with lower risk of ratio of death and lower infection fatality rate ratios. And they say this is consistent with the results from a mouse study showing that blockade of beta alone does not alter the early dissemination of lymphocytic choriomeningitis virus. So that's interesting. So beta doesn't seem to matter, right? It's less important for at least this virus.
For this virus, which is interesting also given the way that we do a lot of immunology experiments and which of the two we measure.
And for in humans, SARS-CoV-2, and in mice, lymphocytic choriomeningitis virus. So they say, despite lower prevalence of these autoantibodies in younger than older, the much higher infection fatality ratio observed in people with the autoantibodies suggests that testing of infected individuals in all age group is warranted. I guess testing for the antibodies, that's what they mean there, right?
Mm-hmm.
And then if you are making autoantibodies, they say we could give monoclonals, new antiviral drugs, or interferon beta when you don't have autoantibodies to beta. They also say that screening uninfected elderly people could be considered because we have 4% of people over 70. You might have some of these autoantibodies, Rich.
Some of your autoantibodies might be autoantibodies to interferon, I think is the way to say that.
Yeah, it sounds like I'm likely to have autoantibodies of one sort or another running around.
Yeah.
Brianne, okay, this screening that goes on is imperfect.
Oh, absolutely. Because if you were going to look for autoantibodies, you would have to have an ELISA or something that looked at all possible autoantigens, which is a bit of a challenge to do. There are a couple of tests that people often will use for autoantibodies, but they really don't include all potential antigens.
Yeah.
Well, what I'm complaining about is that our immune systems are not perfect when it comes to getting rid of all the self-reactive antibodies.
Oh, no. They are very far from perfect in getting rid of both the self-reactive antibodies and the self-reactive T cells.
Not only that, this is an ongoing process, right?
It is.
I mean, if I don't have autoantibodies to something today, I could tomorrow, right?
You could. That's exactly right.
I found this paper about infection fatality rate. It's actually pretty cool. It's is it 2021? So sometime, I don't know exactly the date. This really frustrates me. Some of these articles, it's really tough to find the publication date. Oh, here we go. November 2020. No, published in November 2020. Okay. Let me find we used age-specific COVID-19 associated death data from 45 countries and the results of 22 seroprevalence studies to investigate the consistency of infection and fatality patterns across multiple countries. And then, like I said, there's also math involved. And then they have a bunch of figures here that come up with infection fatality rates. We probably ought to do this paper.
Okay.
It's pretty cool.
All right. So for continuing, maybe we should screen elderly people. And if you have autoantibodies, you should definitely be vaccinated against SARS-CoV-2. And you probably should not get attenuated vaccines like yellow fever vaccines.
So why is that?
I don't know. It's a good question.
Couldn't figure that out.
Yeah, I mean, I guess the idea would be that perhaps those live attenuated vaccines would replicate more in you and would be more likely to cause some pathology. But I would have expected that we would have known that. Although I guess we don't necessarily vaccinate older individuals. We tend to vaccinate kids who maybe haven't developed these autoantibodies yet.
Well, if you're traveling to a yellow fever area and you're old, you would get the yellow fever vaccine. And you probably shouldn't if you have autoantibodies.
They don't let, at least here, they don't let you get it over age 60.
Oh, yeah?
Yeah.
What do you get?
You don't get yellow fever virus vaccine.
Oh, so you go unprotected to, well, I didn't know that. But here's what I think. So 1 in 1.5 million kids who get OPV, oral polio vaccine, the attenuated polio vaccine, get paralyzed. I bet they have something like this, either autoantibodies or some inborn error of interferon. And that's what, and in fact, I bet the 1 in 200 people who get polio, right? Most people who get polio virus infected do not develop paralysis. Only 1 in 100 or so. I bet they have some interferon issue. Nobody's looked because, well, it's almost eradicated.
Yeah, sadly, we didn't have the ability to do some of those things when we were seeing large numbers of polio cases.
I mean, you could take people who have had polio and do these studies still, but
that's true.
But there's less impetus now because, right, it's an eradicated, and people don't.
You might be able to do it for their genetic susceptibility, but probably not based on their autoantibodies at this point.
Yeah, that's right.
Kind of a mess.
Too long, yeah. Well, I'm certainly not going to do it, so please, someone else do it.
I just want to know if I have autoantibodies against interferon.
You probably could assay it in your lab, right?
I probably could.
It's just an ELISA, right?
Yes. I think I only have an interferon beta ELISA right now, but...
They also say, which is an interesting point, it's unclear whether these autoantibodies impair antibody response to vaccines or whether a vaccine-triggered antibody response can overcome type 1 interferon deficiency. Very interesting. And what about other viral diseases? That would be interesting. What are the role of these autoantibodies in other viral diseases?
Yeah, I was asking that before. I was interested in like EB virus, which is very common, right?
Yep.
Yeah, so if I remember that previous paper, none of these patients had been diagnosed before their severe COVID infection with the anti type 1 interferon antibodies. So in that sense, you might say, well, they perhaps did not have terribly serious other types of infections. So I don't recall them having any other susceptibility to other infection from that paper, but it was a while ago.
So they conclude that autoantibodies against interferon type 1 should be a leading common predictor of life-threatening COVID after age and more predictive than sex, comorbidities, and common genetic variants. So we have age, autoantibodies to interferon, and then everything else. So I like this, that it starts to clarify risk factors, right? Age, autoantibodies to interferon. It's cool. And so did we do a paper last week on, what was last week? Oh, last week was inflammasome activation and severe COVID, right? Yeah.
Mm-hmm.
Okay.
I'm actually looking at that other paper right now. They say that before their work, there was one patient who was known to have anti-interferon autoantibodies and severe chickenpox.
And so last week's paper, remember, inflammasome activation in macrophages drives severe COVID, and they speculated that that could be caused by poor interferon responses. And I bet having antibodies would also contribute to that. You can't control infection, and you get over replication, and then you have this inflammasome activation, et cetera, et cetera. I'm starting to understand this. Is that a bad sign?
No, that's a good sign.
I'm not. I mean, you have to be careful because you could be fooled, right?
Well, of course.
Anyway, I like this. This is the kind of science we've been asking for a while. Everybody good?
I'm good.
Let's do some
Yep.
email. Kathy, now we get to do email.
Okay.
Let's see who is the appropriate person, because there's one here that needs to go to Rich. Oh yeah, the second one should go to Rich. Why don't Kathy, you take that first one?
Mike writes, Dear TWiV, thanks for your tireless efforts to educate clinicians and the public on virology and the SARS-CoV-2 pandemic in particular. I've been a faithful listener since the start of the pandemic, and this is my second time writing into your show. I just finished listening to episode of TWiV 898, Immunity is Skin Deep, and like Vincent warned, might happen to listeners, I found myself more confused about the concept of herd immunity discussed in the article they reviewed. The concept of classical herd immunity may not apply. The concept of classical herd immunity may not apply to COVID-19. Thus, after the show ended, I reread about herd immunity, and I believe I came to many of the same conclusions that the host did as to what may have been confusing about the article. Kathy noted the article title used the general concept of classical herd immunity, whereas the article primarily focuses on a subset of that concept, the disadvantage of targeting a certain herd immunity threshold mathematical model to control the COVID-19 pandemic. One might agree with the conclusion of the authors that we've not found a specific mathematical immunity threshold that we can rely on to predict success in controlling the COVID-19 pandemic without discounting the larger and more complex concept of herd immunity. In addition, as Vincent mentioned, with the widespread use of PCR technology, we are measuring control of infection differently. Historically, control meant a decrease in symptomatic cases of a disease. With PCR technology, we are picking up many more subclinical cases, along with some that are asymptomatic. With the amount of times the TWiV hosts have mentioned the possible role of memory and T-cell immunity in the natural or vaccine response to SARS-CoV-2 infection, I'm not yet willing to give up on the concept of classical herd immunity being useful in helping to control the COVID-19 pandemic. I'm waiting to see whether the culmination of natural infections and our vaccine efforts in this community mitigate COVID-19 related deaths and hospitalizations to reserve judgment on whether the concept of classical herd immunity applies to COVID-19. In case other listeners may be interested, I found the following review articles on herd immunity and immune threshold by Dr. Paul Fine very helpful. And he's got the link there to a free article and one behind a paywall. Keep up the great work. Sincerely, Mike.
Cool.
The one that he lists that's behind the paywall, if anyone is wondering, you can actually get it from Google Scholar, not through a paywall.
Oh,
cool. Very good. Thank you. That's good. And this reminds me that yesterday, a couple of listeners showed up, and it was, they walk in and they say, hello, Professor so-and-so, and you're like, who are you? I know you. We're listeners. And there are two pharmacists from San Francisco who we know, and they said, not only do you give us the information we need, but you're part of our family. We feel you are all part of our family. I thought that was so nice, and they meant all of the hosts of all of our programs, right? We've made this family, and people identify with us. I think that's really cool.
They were really enthusiastic. I really enjoyed them, and they
They
also
were
made
really cool.
They also made the point over and over again that it doesn't stop with them. And several other listeners have said this as well, that they are repeaters for TWiV information. They go around and spread it to other people. So it's good.
Rich, you should take the next one, please.
Dear Rich, Vince, Kathy, and Brianne, thanks so much for... okay. This is good. Thanks so much. This is Jeff writes. This is Jeff Smith, who is the senior author on the paper about vaccination that we did a couple of weeks ago. Thanks so much for picking and discussing our paper on smallpox vaccination /microbiota for TWiV this week. You did a great job, and as I listened, you made me chuckle more than once. As this project went along, we hoped very much that there might be a difference in protection following immunization in the presence versus the absence of bacteria. But as you summarized nicely, at least in this model, it was a mouse model, there was not. Of course, this challenge model is a pretty blunt instrument, and the level of immunity following infection by vaccinia virus is so good, getting differences in protection can be difficult. But who knows, as immunity wanes, or in different models, the antibody differences might translate into something. I loved the comments about the third reviewer. And we thought that one of the experiments that was there at the end sounded like it was in response to a third reviewer. Actually, we had 4, and none of them suggested the reconstitution experiment because we did it before submission. So we got you on that one. And how prophetic you were in your comments about testing whether different vaccinia mutants lacking specific immunomodulators might differ in their ability to induce this bacterial expansion. By remarkable coincidence, I was pondering whether to listen to TWiV or to submit a paper on exactly that. Of course, I picked TWiV, and then heard you predict what we might do. Amazing. You certainly were bang on with that one. Okay, I get it. No more per se's. We griped about per se. The link to TWiV was so appropriate. This was the it's a Nature paper, right? By F.W. TWiV, describing bacteriophage. He didn't quite know what he was working with, but it was one of the first observations of what we now know as bacteriophage. The link to TWiV was so appropriate. Thinking back, it was a good job that he, like Fleming, didn't chuck out the agar plates too quickly, and both were observant. Rich also doesn't chuck out his 6-well plates. I remember that photograph of him and a huge wall of 6-well plates. We all love plaque assays. Actually, that was Vincent's wall of polio.
Yeah.
Okay. Me standing in front of it. When you mentioned Peyton Rouse and the discovery of Rouse sarcoma virus in 1911, I recalled that it took the Nobel Committee 55 years to reward him the Nobel Prize for it. Damn good job. He did that experiment early in life. Forgive this long message, but we all love TWiV and send our congratulations and warm wishes that TWiV and all of you and you all will long endure. Best personal regards from Cambridge, England, Jeff Smith. Great. Thanks, Jeff. Good.
Very good. Nice to hear it. Dixon, can you take the next one?
Certainly. Richard writes, Dear TWiV team, your mention of smallpox in the last episode made me think of the recent case of monkeypox in London. As Rich said, pox gets his attention. Could he and the rest of the team say whether or not we should be worried? How contagious is it? And how different is it from other pox viruses? Many thanks, Rich, from Sevenoaks near London. P.S. I like the reference to Fred Twerp. That made me think of previous talk of putting together a list of classic virology papers. That was Vincent's job, and a great job was done there. Well, was this ever done? Of course. You should go back and listen. It would be very interesting to see this in one place, although the references and principles of virology are very good.
Did we do that?
No.
I don't know.
Didn't you do some picks?
You did 10 picks of the
that's
week
right.
on 10 successive weeks of classic virology papers.
They weren't all virology papers, were
Yeah,
they?
there were a few immunology thrown in there, yeah.
Years ago, when I was in graduate school, there was a book that was classic papers in biochemical genetics. Okay?
Mm-hmm.
That was really cool because it had reprints of the papers themselves, and I forget what they were. It's like the Michelson-Stahl experiment and stuff like that. And probably, a beta-galactosidase induction experiment from Jacob and Minot, those kind of classics. Yeah. So, and the way this book was formatted was that it had the actual reprint of each of the papers. And then it had basically an essay, not very long, that described the context of the time, what inspired the experiments, what they learned from it, and maybe a little bit about the authors. And so put the whole thing in context. And to have for a while, years ago, I suggested that there should be a similar thing for virology papers. But,
yeah, You could do it.
It's one of those shoulds. Yeah.
Yeah. So with immunology, in the Journal of Immunology, I think it's once a month or so, there is something called the Pillars of Immunology series, where they reprint a classic paper in immunology and have a little perspective piece that accompanies it that does something similar. And I have found some of those fascinating in terms of giving me insights and explanations on some classic experiments. So I wonder if something like Journal of Virology could do something
When
similar.
I was on the editorial board of Virology, I proposed doing this. But this is one of these things where, if you're going to propose something like that, you better be ready to do it yourself. And what I've discovered over time is that if you're going to write stuff like that, you have to be one of these people who likes to write.
Yeah.
And that's not me.
That's right. I forgot. You're right. You like to talk, though.
I like to talk.
Okay.
Talking is fine.
We got you here.
Writing is another thing.
Excellent. But anyway, Rich from Seven Oaks, we talked about it. So I think we covered all your questions, right?
Talked about the monkeypox, yeah.
Brianne.
Lowell writes, Vincent et al., I appreciated your discussion of the herd immunity to SARS-CoV-2 paper by Morans et al., and particularly their and your mention of the 1917 paper by Eichhorn and Potter, where veterinarians were ahead of the MDs. I immediately looked up the paper, and it is a great reminder that folks were smarter than we think back then. It is attached for Brianne's convenience. Thank you, Lowell. Vis-a-vis the mouse paper, another potential example of vets being ahead of MDs is the attached Troxel et al. Paper, wherein they reported that cattle vaccinated against Clostridial disease that developed injection site lesions had marginally higher titers, generally in the 2x range, with p-values ranging from 0.02 to non-significant, than cattle that did not develop lesions. See Table 1. Notes: Clostridial vaccines for cattle are notorious for leaving lumps, ranging from inflammation that eventually resolves completely to granulomas to abscesses. My suspicion is that the vaccine maker funded the study so that they could tell producers to not worry about the lumps. It just means that the vaccine is working. Still, folks with show cattle hate the lumps. Clostridial 7-way vaccines for cattle are bacterian toxoids, with the toxoid part being more important, see below, against Clostridium chovoi, blackleg, septicum, malignant edema, novi, black disease, sordelli, and perfringens type C and D, enterotoxemia. Note that there are only 6 pathogens mentioned. The toxoids in the C. perfringens type C and D provide cross-protection against type B toxins, so type B is the secret 7th in the 7-way. If you read the rest of the paper, the following might be relevant. Experiment 3 involved vaccination in the face of maternal antibodies, IFOMA. The waning of maternal antibody is variable, but since you got to pick a time to start a vaccination program, 3 months of age, 4 is better, is generally the magic number chosen for when most folks consider maternal antibody to have waned in most calves on most farms most of the time. Experiment 1 weaned at 8 months old, and Experiment 2, stocker heifers, age not specified, but likely greater than 3 months based upon weight, which is specified, were performed at greater than 3 months. When I used to give the lectures on Clostridial diseases in cattle to the vet students at the OSU, I'd make them repeat after me, Clostridial disease is all about the toxin, then make them repeat it until they were enthusiastically shouting it. Then tell them that would be on the exam, then put it on the exam. I've been busy and not keeping up on TWiV as much as I'd like, but I'm glad I caught this one because you never disappoint. Warmest regards from Lowell, and Lowell is a vet from Merck.
Cool.
I'm glad we don't disappoint. Thank you, Lowell. Good to know. I think this is a great strategy. I often do this. I don't make the students shout, but I'll give them a question and then put it on the exam. And the ones who get it wrong, they didn't pay attention in class, right?
Every year, I tell the students when I talk about one particular thing in viral transcription, DNA viral transcription, I say, every year I tell the students that I'm going to put this on the exam, and I do. And every year some students get it wrong.
Yeah.
And I predict that will happen this year, and it always does.
Yeah, it's very interesting. It's very interesting. All right, John writes, well, 898 certainly dovetailed nicely with a couple of things I was about to pass along. The first is a polio connection to the design of one particular automobile with an aftermath in vaccinology. I was recently reminded that Alan Leamy, who is generally credited with designing the Model J Duesenberg, which most would agree is one of the greatest designs in automotive history, contracted polio at an early age and walked with a brace and cane for the rest of his short life. Could his impaired mobility have contributed to his gravitating to a desk job in design? He provides a link for that. Unfortunately, he also designed the 1934 Auburns, which didn't sell well, and so he was let go from Auburn Cord Duesenberg. Go see the ACD Museum in Auburn, Indiana, far northeast corner in the old factory showroom. It is absolutely wonderful. He was almost immediately picked up by GM for the LaSalle Junior Cadillac Design Studio, but this was tragically unfortunate since he came down with sepsis after routine diphtheria vaccination required by GM. So either that dovetails with any immunization is going to introduce some bacteria into the muzzle, as Vincent commented at about 1/11/11 and 898, or the vaccine preparation contained live prophage-infected Corynebacterium diphtheriae. Wow. That's very interesting. And in parens, the mechanism of action of diphtheria toxin is insidious. Super briefly, the toxin enters the cell after docking with a heparin-binding membrane protein, followed by endocytosis that then enables the toxin A subunit, an ADP ribosyl transferase, to modify an already modified histidine residue in elongation factor 2, the translational machinery, causing a steric clash that blocks all protein synthesis and leads to tissue necrosis.
You can't make this stuff up. It's crazy.
The other aspect of 898, generally about other factors affecting the immune response, tied to a question I had recently after having gotten over an almost 2-week bout of hay fever. This came immediately after a gathering of about 600 for the combined 50-year reunion of 3 classes of William and Mary grads. The last 2 years' reunion were postponed by COVID. In the absence of any vaccines, this would have been a super spreader event, so I was a little nervous that it wasn't simply hay fever. I'm triply Moderna vaccinated and tested negative, but still, because of all this, I started to wonder if any of the cytokine response to allergens might have a beneficial effect versus concomitant exposure to SARS-CoV-2. I found this that seems to speak in that direction, but I'm sure that Brianne will have a much easier time understanding it than I do. And what do we got here?
It's a link to a paper in the Journal of Leukocyte Biology from 2015 that talks about interferon in allergy and viral infection, and that I will read later today.
So the idea is that if you have an allergic response, sometimes it could help you with a viral infection, right?
Right.
Because of the cytokines that are produced, is that right?
I think that's the idea.
Okay.
As someone who has an awful lot of allergic responses, I sure hope so.
Are you allergic to your cat?
I am not allergic to my cat. I'm mostly just allergic to outdoors.
Well, then you should stay inside and podcast on Fridays.
Exactly.
So far as I'm aware, none of those attending, many of whom were quadruply vaccinated, tested positive in the aftermath. So we may have participated in a large, uncontrolled experiment in vaccine efficacy, herd immunity, but I guess it really just amounts to a large-scale anecdotal evidence. Indeed. Thank you, John, for getting it. Otherwise, just a splendid, perfect day here in Greater Braddock today that started off at 4C after several days of monsoon and reached 20 without any clouds or wind. Your results may have been similar. We won't have many such days here. This was the first of 2022. Best regards, John. Wow.
Greater Braddock?
I don't know where Greater Braddock is. We could find it. Greater Braddock.
British Isles?
Pennsylvania.
Pennsylvania.
There you go.
Yeah, I see the Monongahela River. Doesn't that go through
All right.
Pennsylvania, right?
Yep.
Yeah, Greater Braddock, Chamber of Commerce, Pennsylvania. And of course, this could be a fake website I've reached. That's, I just read the Death of Expertise, and that's a whole chapter devoted to don't believe the first website you hit.
I have independent confirmation
and that's
from Google Maps that results in a whole bunch of hits outside of Pittsburgh.
Okay,
or
yeah,
actually around Pittsburgh.
All right, let's do another round. I think this would be fun. So I just finished, and who went first? Kathy went first, so it's your turn. Yeah.
Robert writes, "Thank you for sending me on a rabbit hole of smallpox history. I thought to share a bit of info I found. It was discouraged to use alcohol before pricking with the bifurcated needle due to a fear of the alcohol inactivating the live virus vaccine. This is noted on both the DryVax package insert and the ACAM 2000 insert. No skin preparation should be performed unless the skin at the intended site of vaccination is obviously dirty, in which case an alcohol swab may be used to clean the area. If alcohol is used, the skin must be allowed to dry thoroughly to prevent inactivation of the live vaccine virus by the alcohol. It is also noted on the CDC website, which also has an interesting instructional video series on administering it as well. I do not know if this changes any conclusions, but I found it intriguing. Thank you also for the book recommendation for The Love of Enzymes. Here's to hoping it gives some additional inspiration to changing careers."
I didn't know that. Did you know that, Rich Conn?
No, we this came up during that discussion.
Yeah.
And Robert has either independently or picked up on something, and I think I mentioned the other day, is that when I want to know something about a vaccine, I look up the package insert.
Mm-hmm.
Okay, because that's it's formatted. So I know how to read these things. I know where to look for what I want to look for. And it's really a nice synopsis of all of the stuff that you might want to know about a particular vaccine. So it's really a great resource. And as he talks about the YouTube videos, I've seen instructional videos on vaccinating, or talk to people, I forget which it is, where they practice on an orange. The skin of an orange being, a readily accessible material that's, similar to skin of a human, I guess, for practicing how to use your bifurcated needle.
Rich, you're next.
Cyrus writes, "I'm a student in the BE 115 Virology class at Caltech. As part of our coursework, we were encouraged to reach out to TWiV with questions for extra credit. My question is on your TWiV 898 episode, Immunity is Skin Deep. In the episode, the panel discussed a publication that suggested herd immunity, a concept originating from veterinarians during World War I, was ineffective for COVID-19. The point was made that while the concept still applies to this disease, the evidence gathered during the pandemic indicates that herd immunity is ineffective at controlling COVID-19 infection rates. While some viruses, such as measles, are effectively captured by herd immunity, the panel concluded that there may be some viruses that herd immunity doesn't apply to due to the antigenic effects and population dynamics. My question to the panel is this: is there a way to predict a priori which viruses herd immunity will be applicable, i.e. key viral genetic indicators, typing, or compatibility? Does the herd immunity model have underlying assumptions that precludes its application broadly to all viruses? So, good question. Okay. The bottom line, good question, which is the scientist's way of saying, I don't know. But I want to start off by saying that I think we're still confused about how to use the words herd immunity. I think we probably shouldn't use them at all, okay? Because it has become a term that is misunderstood in lots of ways. I talk about population level immunity or something like that, something else, because I think it's pretty clear that the immune status of the population in the United States is having a profound impact on our experience with SARS-CoV-2. Okay? In particular with disease. And I don't think we know yet what the impact is on the infection. I doubt very much if population level immunity, as we're experiencing it now, has no effect on the infection rate. Okay? My guess is that it's going down. The problem is that early on in this whole thing, the concept of herd immunity was presented in many sources as sort of a black and white concept, that when we reach 70%, this is all going to go away. All right?
That's right.
And I think that those in the know, even at that time, would have said, well, I doubt very much if the virus is going to disappear, but the infection rate in the population will be low enough so that it's no longer a problem. Well, you know, what number is that? I don't know. But so it's a squishy concept. That's the problem, and I think it's misunderstood. But I think from your letter, Cyrus, I think you understand the fundamentals. Now, your question is really interesting, okay? Because clearly, a virus circulating through a population, this concept of what impact does population level immunity have on the virus, and how does that change depending on what virus it is and what the characteristics of the virus are. Frankly, I don't know that there's a really good answer for that. I think it's a very, very complicated question. Depends on how it's transmitted, how efficiently it's transmitted, whether there's asymptomatic transmission, what the infection fatality rate is, how serious the disease is, all these kinds of things.
Yeah, well, the other thing that becomes really important here, you mentioned exactly as I was kind of thinking, Rich, that has always been this idea of herd immunity as a black and white thing. Either we're at the magic number or we're not. And I think I think herd immunity sort of makes this assumption that immunity is also a black and white issue. You're either totally immune or you're totally not.
Right.
And I think we know that that's maybe not true, in that you may have, partially effective immune responses due to antigenic change. You may have immune responses that last for a longer or shorter period of time. And so it's not as if we can mark every person as perfectly immune or perfectly susceptible. And so I think that gets to that same squishiness that you're mentioning.
And it's also true, is it not, that your immune response to different viruses is different. Some viruses induce a longer lasting immunity. Other viruses don't. Some viruses are more prone to antigenic shift or drift than other viruses. So I think this is going to be a case-by-case thing. And I don't know, I mean, it would be interesting. I don't know I can't think of any resources where there might be some sort of, where somebody tries to lay down the fundamentals of the population immunity with respect to the type of infection that you were talking about.
I've seen a little bit of it, and it gets well beyond my math skills quickly.
There was the one publication from NIH, John
Moorens, David Moorens?
No.
John Udell.
John Udell. Yeah.
Sorry. Where he was making a point about the immunity to respiratory viruses versus viruses that establish a viremia. Okay?
Yeah.
And I think the durability of immunity, okay? And now that's sort of a step back from talking about herd immunity, but that's a sort of a general concept, okay, that might play into population immunity. Okay, so Cyrus, keep on going. All right?
Exactly.
And you've come up with a great question here, and you're going to be the guy who ultimately tries to flesh out some fundamentals of the population immunity dynamics to different virus infections. How about that?
So this,
And Richard, it's not just about viral infections either, because
yeah, of
exactly.
course,
Yep.
of course.
Malaria is a great example of a seasonal infection that comes back and infects the same people again and again. They get less sick each time, but each infection, particularly with Plasmodium falciparum, which is the most dangerous of the 4 species that infects humans, has a dramatic effect on the cognitive development of children. So I'm curious as to know whether all you virologists out there, rather than you carriologists like myself, have an opinion as to whether or not the concept of herd immunity might have begun when the effectiveness of the smallpox vaccine and yellow fever vaccines were such that if you give it once, it's good, and you can head off a disease this way. In fact, you can head off an infection this way, right? So maybe the old guard of smallpox vaccine development and administration too, because the hope was that if you vaccinated a certain number of people, you could wipe it out because it couldn't transmit anymore. I don't think they had a number in mind, but I used to hear lots of talks at the American Society for Tropical Medicine and Hygiene by these people like Soper and lots of other old time, you'd consider these old school first generation immunoepidemiologists that are just trying to eradicate everything. So start with the simple ones, and the simple ones look like yellow fever and smallpox because we had vaccines. So we can create herd immunity, and the rest is history. But look at how complicated life got the moment we encountered SARS-CoV-2 and then malaria. Of course, malaria was always around, but that was not a virus. People weren't thinking about herd immunity at that point, but they certainly were with regards to viruses. So you mentioned you hit all the right buttons. I just sat here, a non-virologist, thinking about this. Rich hit every one of those things, and it's too bad I'm just talking too much about the same thing because you've already said everything I wanted to say, and darn it, I didn't get a chance to say it first.
I just wanted to say something about prediction. So Cyrus asks if we can predict. And so I read, as I said, The Death of Expertise, and there's this great line about prediction. Let me read it to you. Prediction is a problem for experts. It's what the public wants, but experts usually aren't very good at it. This is because they're not supposed to be good at it. The purpose of science is to explain, not to predict. And yet, predictions like cross-expertise transgression are catnip to experts. So it made me very sensitive, which we do all the time. We try and predict stuff. We shouldn't be. But I also like the cross-expertise transgression, e.g. stay in your lane, right?
Well, so as long as we're at least mildly digressing here, this reminds me of the conversation that happened last night at the party, where the question came up of why was there not a coherent picture/policy communicated from some central resource to the population early on in the pandemic, okay? So it wouldn't be so confusing and we'd all be on board and know what to do all the time. And, one of the things that didn't get discussed in response to that question is it's because we didn't know what was going on, okay? People did the best they could with the information they had at the time, which was incomplete. It was not a new pathogen, though not entirely unknown pathogen, but certainly in an unknown circumstance coming into a naive population. And our perspective on it changed over time. Plus, the dynamics of the disease in the population change over time as the population acquires immunity and is still changing. Okay, so I get a little frustrated when I hear people talk about how the communications, in particular from various government agencies, are confusing. Okay, it would be irresponsible to try and give a consistent story when the reality is not consistent. Mm-hmm. Okay, things change. Get used to it. Deal with it.
They do. I think the public's expectation for predictability from science began in the early 1900s when the general and the special theories of relativity were first emerging from the physicists, and they would make predictions about, whether or not if an occult event occurred where a star passed in front of another star and then it sort of moved away from that, would the light bend around the first star or not? And then how much bending would that occur? And based on their hypotheses and their mathematics, Einstein was really vindicated in a lot of his early predictions as to what his theories actually meant. Now, and then you compare that with biology, even regular chemistry. There's so many other variables that go on in larger systems where you don't have a good, tight mathematical model to start with. So there was no good, tight mathematical model for herd immunity, none. If there were, I would have heard about that, no pun intended, but I would have had that in graduate school. They would have said, okay, here are the parameters under which you must consider herd immunity with regards to vaccination for, and it shouldn't really matter whether it's a virus or a bacteria or a protozoan or even a helminth. But in physics, you always got Einstein's predictions, always. And over time, I think the only one that he didn't succeed at was predicting black holes, if I'm mistaken, but I probably am. But that's the kind of precision that the public expects. And where does it start to all fall apart? And none of us have a good handle on all of the parameters that you would need in order to include them in an airtight mathematical model with regards to herd immunity. And I've never seen such an attempt after the early attempts failed.
Well, I think the other thing to point out that goes with what both Rich and Dixon are saying is Dixon mentioned some of these pieces of predictions that have been borne out by Einstein's work. And one of the things you mentioned was that happened over time. Because there is often some time between when we make an observation and we make a bunch of observations, we put them together and we start to have an understanding of the picture. And I think that what was frustrating to me during a lot of this was the public sort of seeing an observation in almost real time. At the same time, the scientists were seeing it and sort of asking, well, what does it mean? Tell us all about it. Tell us the background. And I know that I always felt like sometimes saying, I just found out 2 minutes ago too.
Right.
We need the time to actually process this information and make the observations.
Yeah, it's a digital age, right? We're all sort of tuned into this 24-hour news cycle, and our attention spans are 15 seconds long.
Well, the science is not attuned to a 24-hour news cycle.
That's right.
It takes time, as Brianne said. You have to think and contemplate, sit back, and they want an answer. What does it mean immediately? I have a deadline in 10 minutes. It doesn't work. By the way, Dixon, before the pandemic, I used to teach there is a formula for relating R naught to the fraction of immunity you need to get herd immunity. I have since COVID threw that out the window for me.
Exactly. And probably a lot of other infections too that we didn't care about because they were so minor, like the common cold. Some people get it a lot and some people don't get it at all. And there's how many strains of rhinoviruses out there, right? So,
but to quote a famous movie, it's not a law, it's more of a guideline.
That's right.
I'm going to take us in a different direction, but everybody's gotten to digress on Cyrus's letter. I went off on the fact that he's taking virology at Caltech, and at least in 2020, and I think now, it was taught by Pamela Bjorkman.
Yep.
And I got to hear her talk last week at our Virology one-day symposium here, and it was great. Those of you who are going to ASV 2022 will get to hear her give a plenary talk at Madison. But the funny thing is, to kind of confirm this, I went online to look for this course, BE 115, and the page I landed on said, "You don't smell like a human." And then it's a bunch of little robot figures, and then you click on it, and it takes you to a CAPTCHA page to verify that you're not a robot. But it was really funny. So.
And I can tell when she's teaching her course, because I get a flood of these emails, all about the same time of year. Yeah. All right. There was nothing else I wanted to digress on. Dixon, what do you have for us this week?
Well, I read something very interesting. I go to the NASA website a lot. I'm sure Rich does also. Probably you all do. But they just recently decided to see whether or not they could germinate plants in regolith. And regolith is moon dust. And it's crushed rock, basically. It's just like meteorites hitting the surface of the moon and pulverizing it into a powder, which is what I was concerned about the lunar lander module and the first touchdown. How deep into the soil would that actually go? What is the regolith? And it turned out to be very thin, by the way. They germinated a plant in it by just adding a few more nutrients. They need 16 elements altogether, and it turns out that you can now use moon soil with a few added elements to grow plants. And they've already discovered water on the moon. So you've got all the elements there for a permanent moon colony that can raise its own food, whether it's in a vertical or not configuration, who cares? But it looks like it's a colonizable unit because of that, and I wonder how fertile the soils of Mars will be.
Cool.
That is
I'm
very
thinking
cool.
of Kate.
Tiny little plant they show here.
I'm thinking of Kate. I'm also thinking of The Martian, right?
Yep.
Well, that was a little added.
Yeah, he had a little added stuff in it. But, if humans go to the moon, there'll be a little added. So, and this interview that you picked up is with scientists from the University of Florida.
Yeah.
Yes, they were. Yeah, indeed, they were. Indeed,
they include Rob Furl, who's an acquaintance of mine. Cool.
Indeed, they were.
Small world. Thank you, Dixon. Brianne, what do you have for us?
So I have a recent astronomy picture of the day. It's from May 16th. And this one really stood out to me. I really enjoyed it. And for some reason, that day, I kept trying to open browser windows because I wanted to see this over and over again.
Wow.
It's called Milky Way over French Alp hoodoos. And so you can see the Milky Way here, but you also can see some really interesting geologic formations that are found in France. And so I learned about hoodoos that include dense rocks sort of slowing the erosion. It looks a little bit like a castle that's found in the French Alps. And then you can see, so you got these really cool sort of castle-like rock formations with the Milky Way galaxy behind. I enjoyed this one a lot.
Brianne, what would this look like if they superimposed pillars of the gods in the back of that for all of the star formation in these big, gigantic collections of stardust?
That would be cool.
I think there's a huge connection that's going on here somewhere, and this is fabulous. How would you like to wake up camping and see that?
Have any of you been to Bryce Canyon?
I have the page up with the images of hoodoos at Bryce Canyon.
Because that is like, the hoodoo, it's a hoodoo palace. Yeah. And my recollection of the story in Bryce Canyon is that the Native Americans at the time, I don't know if this has anything to do with the derivation of the word hoodoo, but the Native Americans at the time, their story about the hoodoos is that there used to be a population of bad humans who lived in the area. And the tricky coyote didn't like it, and so he turned them all to stone. And that's where the hoodoos came from. I really love it.
That's nice. Wow. Oh, I see some of these hoodoo pictures from Bryce Canyon. Those are cool. Nice. Now I'm going to have to make sure I go there.
Oh, yeah. Bryce Canyon is spectacular.
Yeah.
It is.
Cool. Kathy, what do you have for us?
I have a data visualization, one more for COVID-19. It's nationwide. I actually didn't explore it to see if it goes further than the US, but it's got some really nice maps, and you can scroll down, you can zoom in. And this is another thing from our listener, Justin, but I find it a really good data visualization page. And at the very top, they say, SARS-CoV-2 is airborne. Wear a KN95 plus. Get vaccinated and boosted. So.
This is really detailed.
Message. Yeah.
It is very good.
Especially those with one interferon anti-autoantibodies.
Indeed.
This looks cool, Kathy.
Rich, what do you have for us?
I just want to plug a research program at the NIH called All of Us. And it's, there have been programs around like this, but this is the first of its kind, I think, that's, funded by the government or that's housed in the government. And this is an effort to collect genomic and other health data from at least 1 million individuals and keep going with the goal in mind of, detecting, correlating health profiles and genetic information with an eye on the long run of, developing approaches to personalized medicine. And so you can actually join the program if you want, send them some spit and fill out some forms and become part of their database. I know about this, among other things, because my brother-in-law, Jeff Ginsburg, has been hired. I had it here for a while now. I got so many tabs up, I lost it. Is the scientific director, I think.
So they partner with various institutions, and Columbia is one of their partners. So I did this last year. Went and they take various fluids from you, blood, urine, saliva, maybe. They take a little questionnaire, and then you never hear from them again, but it's good. My
Jeff is the Chief Medical and Scientific Officer.
Cool.
Okay, so
nice.
Good luck with that. I mean, this is where medicine should go.
Yeah, no, I think it's great. So my pick is a perspective in the New England Journal of Medicine, which unfortunately is behind a paywall. It really should not New England Journal of Medicine. I mean, if you want people to think about things like this, what are you charging them for? What is the point of that? I know what your model is, but now and then you have an article like that which addresses something that many people should think about. It shouldn't be behind a paywall. It's called Physicians Spreading Misinformation on Social Media. Do right and wrong answers still exist in medicine? By Richard Barron and Yul Hines, both doctors. And it begins by saying medicine has a truth problem. In the era of social media and heavily politicized science, truth is increasingly crowdsourced. The wisdom of crowds, which has particular power in a democratic society. I want to point out that a democracy means simply that everybody has a vote. It doesn't mean everybody's right.
Correct.
Okay, remember that. That's very important. And so this is all about physicians who go on social media and say things that are not right. Medical misinformation. I'm not naming names. I don't want to be sued.
No, I don't name names in this case. He's running for some office in Pennsylvania. How could people possibly want him to guide them when he's incredibly disruptive and misleading on purpose?
Well, he doesn't think he is. That's part of the problem, right?
Yeah, well, he knows. No, that's not true. I've actually talked with him.
Yeah?
He knows what he's doing.
Well, anyway, I want to summarize this article because if you can't get to it, I want to summarize it. So basically, how do we deal with assertions by doctors that are not supported by evidence and could be harmful? Medical misinformation, they say, is a major public health threat. And there's a very interesting issue here. An individual has questioned, for example, why First Amendment protections should extend to harmful speech that leads to death from disease when other forms of speech, such as fraudulent commercial speech, which may be less harmful, are prohibited. So he's questioning why. Why don't we, make this illegal to do that? And so they go on to talk about how medicine is policed. It has certifying boards that make sure that you know what you're supposed to know. There's an American Board of Internal Medicine that makes you pass exams on subspecialties to ensure that doctors are practicing evidence-based medicine, right? And you're driven by the latest peer-reviewed research, which we know doesn't always happen because it doesn't trickle down to everyone. They go on to talk about how when you make up an exam, you always want to have a distracting question, which clearly isn't right. But sometimes it's really hard to make a question that is really totally wrong, right? And so this is part of the problem with physicians making incorrect statements. It's hard to know when, not always, but sometimes it's hard to know when they are incorrect. And so they say a whole range of statements with which many or even most physicians might disagree would therefore not trigger our disciplinary process. So this is part of the problem because they're in that gray zone. So on the other hand, when someone certified by this American Board of Internal Medicine says something like, children can't spread COVID or vaccines don't prevent COVID deaths or hospitalizations, we're not dealing with valid professional disagreement. We are dealing with wrong answers. So that's the point of this. There is professional disagreement and then there are wrong answers. So the last sentence of this is there aren't always right answers, but some answers are clearly wrong. So that is the answer to the question posed in the headline. Do right and wrong answers still exist? Yes. The answer is yes. And so it's very important because, as you know, on social media, there are certain physicians who say things that are wrong. And Dixon named one of them, but there are others as well.
This is particularly true with regards to human nutrition.
Oh, yeah?
You find a lot of physicians taking sides on issues, and you get whole cohorts of physicians saying a high-fat diet is a good thing, and then another one, of course, comes along and says you're going to develop coronary disease if you do that. It's crazy to try to pick out, unless you go to these specific exams where boards have reviewed the literature and they haven't found any evidence for. But the Harvard School of Public Health almost every year comes out with a new take on caffeine. Like 1 cup of coffee will make you more alert and happy. Well, 3 or 4 will make you even better. And then the next time they come out and they say, well, wait a minute, that's not true. And I don't know how that varies unless the head of the nutrition department at Harvard University School of Public Health keeps changing every year. So the new one that comes in has a different opinion than the old one that just went out. It's that variable in many cases. And it's really, I don't know what you do about that because human nutrition.
Dixon, experts are not always right, but they're not always wrong either. So
no,
they're just
I agree
errors.
with you. But the way they say things sometimes, it's like ironclad. Like eggs are good for you.
Well,
no matter how many.
Do you remember when they said they were bad for you?
Yes, of course I do. Of course I do.
And then people started eating other things, and we had an epidemic of obesity.
The cholesterol levels in your blood, the HDLs and LDLs didn't come out for a long time before that data was sorted out. It's still not really sorted out, basically, but.
I just sent the journal a message telling them to make it open.
Don't you think this should be open if
Absolutely.
they want people to think about this, not just doctors, right?
Yeah, absolutely.
Make an article open. Come on, you're not going to lose any money. Give me a break.
You might even recruit new subscriptions.
We have a listener pick from Lisa. Hi, TWivers. I really value TWiV. Thanks for making it. Alan Dove's pick on TWiV 894 reminded me of the biggest tourism highlight of a pair of trips my husband and I took to San Angelo in the spring of 2021, the E.H. Danner Museum of Telephony, housed within Fort Concho. A remarkably large amount of cool telephone technology packed into a very small area. Our trips to San Angelo were virus-related in that's where I got my spike vaxes back when it was really competitive to get one around here. And Lisa is from Austin, Texas.
Cool.
That's very neat. That is where's San Angelo?
I'm just looking it up. I don't know.
Is it in Texas? It must be, right?
I don't know.
She's from Texas.
Yes, but, to get the vaccine, she probably stayed within Texas, no?
Texas.
Yeah, it's in Texas.
Google Maps says it's in Texas.
It is in Texas. And let's see.
It is. Wow, it's out there in, it's sort of northwest of Austin.
Yeah, there you go. It's about 2 hours, you think?
Probably even more than that. Texas is pretty big.
No, 50 miles is an inch. So it's 1, 2, 3, 4 inches. So it's 200 miles. Very good. Thank you, Lisa. Next time I go to Austin, can we go?
Sure.
To this museum?
Yeah.
We have to see Nels's parents' museum.
Yeah.
Just somewhere in Texas as well. All right, that'll do it for TWiV 902. You can find the show notes at microbe.tv/TWiV. You can send us your questions, your comments, your picks of the week, the TWiV at microbe.tv. And if you enjoy our work, we would very much appreciate your financial support. You can go to microbe.tv/contribute. Your contributions are federal U.S. tax deductible. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas, but right now he is right next to me at the incubator in New York City. Thank you, Rich.
Sure enough. Always a good time. And San Angelo is 3.5 hours from Austin.
3.5 hours.
So we will rent the canary yellow Mustang convertible and drive to San Angelo.
So yesterday we were speaking with Jeff, who helped to organize this event yesterday. And he's saying, well, you should come to the West Coast and we'll do a series of these from San Francisco to Los Angeles. I said, fine. I want you to rent the canary yellow Mustang convertible. And he said, I have a white one right in the garage here. So apparently he went to rent a car and they had nothing left except a white convertible Mustang.
Cool.
And this guy is driving it around Manhattan. Can you imagine with the top down? He said he drove through the Lincoln Tunnel with the top down. He's crazy.
Oh.
I hope he held his breath.
But you can tell he's from Los Angeles. He comes to New York and he rents a car.
From Los Angeles, he didn't have to hold his breath.
So anyway, thank you, Rich. Dixon Despommier is at Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're welcome. Rich, I love your tag, so I'll just say it. Always a good time. Ditto. I'll just ditto. If you go before me all this time, I'll just ditto you because it is a good time. It's a fun time. It's actually fun time.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
And Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
What's wrong? What did I do wrong?
Nothing. It's the plural.
Nothing. You put the right
word.
Okay. Because sometimes I say things wrong,
I didn't say anything wrong.
Okay, good.
People are playing with a Google Doc here.
Okay. I'm just focused. I'm just like a laser. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.